biotechnology we learned in the previous chapter is accelerated the industrial scale of production of the biosperm and biopair bio BHRBTudinal production of pharmaceutical and biologically using genetic modification microscope biologically using genetic modification of the microscope fungi plantae fungi plantae animalia this application of the biotechnology include therapeutic therapeutic this application of the biotechnology including therapeutic diagnostic genetical modification of crop food agriculture this process of food process of food bioremediation process of food processed food bioremediation waste treatment and the energy production three three critical research area of the biotechnology they provide the best catalyst for the improvement of organic using microbe and pure enzyme this the correction of optimum condition through the engineering a catalyst act the optimum condition through the engineering for the catalyst to act the downstream the processing the technology purify the protein organic compound. Let us learn the let us now learn the let us now learn the human being have the used the biotechnology to improve the quantity of the human life and especially the field the food production and the health food production and the health biotechnology is application in agriculture they look the three option that can throughout for the increase the food production agrochemical based agriculture agriculture agrochemical based agriculture agriculture and organic agriculture and genetical engineered crop based on agriculture crop crop based on agriculture agrochemical based on the agriculture agrochemical based on the first हो जाएगा agrochemical based on agriculture organic agriculture एक हो जाएगा crop based on agriculture the genetical engineering of the crop based agriculture green revolution success by the tripling of food supply succeed by the tripling of food supply it was not enough to feed the growing the human population It was not enough for food growing. The human population increase yield the part of increase yield have the partly been during the improved the crop variety increase in the increase yield the partly partly been during the improve of the crop variety, but mainly due to the use the better management practice and the agro and used to agrochemical filter and pesticide agrochemical filtered and pesticide. However, for the frame the developing world the agrochemical are often expensive. Further, the increased yield with the extensive variation not possible to use the conservational breeding. Not the developing world, the agrochemical are not often expensive. agrochemical expensive हो जाता है क्योंकि further increase yield with the extensive variety is not possible to use the conservational breeding. The technical a- as tendency breeding of the technology tendency breeding of the technology field keep pace with the demand and proved the and proved su- mm, suff- sufficient fast and if efficient system. Intentional breeding is the technique field क्यों क्योंकि keep pace with the demand with the and provide sufficiently fast भी provide करना था और and the efficiently system the crop improvement के लिए and another technology are called the tissue culture और दूसरा technology है tissue culture developed with the food developed food के साथ करेगा tissue culture means it was the learned by the scientist during the 1950 टिश्यू कल्चर लर्न किया गया साइंटिस्ट के द्वारा कब किया गया था 1950 में that whole plant would be regenerated for the ex, ex from ex plant से any part of the plant take out and grown any part of the plant out and grown in the test tube under the sterile condition and the special nutrient media under the sterile condition special nutrient media the capacity of the generating the whole plant from any cell and explant are called the to to tense totipotence the whole plant from the any cell या फिर एक्स प्लांट इसको बोलते हैं टोटिपोटेंट we learn how to the accomplish accomplish this is a higher classes. It is important to stress here the nutrient medium. It is here to nutrient medium must be provided the carbon source and the carbon source as such source also inorganic salt carbon source and source also the inorganic salt vitamin and amino acid growth regulation like the auxin in auxin auxin and guide to auxin and GY2 sign. The application of this method, it is possible to achieve the progression large number of plant and very short duration large number of plant and very short duration. This method for the production thousand of plant. This method for the production thousand of plant tissue culture is called the 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 macro propagation. Tissue culture is called the macro propagation. Macro propagation. Each of the plant will be genetically identical to organelles genetically identical to the organelles plant from the where grow from they will grow. That is, they are called the सोमिक्लोन क्या बोलना the identical of the organic plant from which they are grew. They are somiclone. Many important food plant like the apple, apple. Has been the produced commercial scale. Using this method, try to visit the tissue culture laboratory. Your teacher is better understand and the appreciate this process. Another important application is the method to recover the healthy recover the healthy plant recover the healthy plant for the diseased plant. Even if the plant is the infected with a various the mercent मेरिस्टम sorry virus क्या बोल रहा हूँ? With the virus and the meristem, the up, up, apical and axillary, the free of the virus. Hence they one or the remove the meristem and grow into vitro. Remove the meristem and grow into the vitro. They obtain the virus-free plant. Scientists succeed to the culture in the meristem of banana, sugarcane, potato etc. The scientist has even the isolated single cell from the plant, and after the digestion their cell wall has been able to isolated digestion their cell wall they are able to isolate it. The necked population the necked protoplast the naked protoplast surrounded by the plasma membrane the isolated protoplasm from the two different variety of a plant proto isolated protoplasm from the two different variety of the plant. Each has the dis in disable character. Each has the disable character. Can you fused get hydrate protoplast for the grow from a new plant. There are the hydrates called the somatic hydrate. They are hydrate called the somatic hydrate, while their poses are called the somatic hydration, which can be further grow from the new plant. These hydrate are called the somatic hydrate, while the process is called the somatic hydration. Process is called the somatic hydration. The image of the situation when the protoplast of the tomato fused with the protoplast of the tomato fused with the potato, and they are grow to form the new hybrid of plant, new hybrid of the plant. And combining tomato potato characteristic, well, this has been achieved, the resulting in the formation of tomato unfertility unfertility बोल कैसे रहा unfertility गधी रही unfertility. The plant did not have the described the combination of the characteristic for the commercial and utilization. Unfertility plant do not have the described combination of the characteristic for the commercial utilization. In there, all the alternative pathway that are the understand of genetic alternative alternative pathway understand the genetic can show that the frame may be obtained maximum yield their field field is there a way to minimize use the filtration chemical um, minimize way to minimize the filtration and chemicals, so there is the harmful effect on the environment reduced. The genetical modification of the crop possible for solution. The plant the plant plant fungi animal who is the genus been has been alerted. नहीं नहीं एक मिनट चलो पढ़ो whose gene whose genus has been altered by the manipulation is called the genetical modification organism. GMO, genetical modification organism. GNO. GM is the plant have been useful in the many ways. Genetical modification, the crop has the tolerant to abiotic tolerant, more tolerant. The crop is more tolerant to abiotic state like cold, mm, dust, salt, heat, and reduce the reliance on the chemical pesticide like pest resistance crop like pest resistance crop. They help in the reduce the post-harvest losses. They increase the efficiency minerals use the plant. They prevent the earlier the exhuming. the filtrate of soil. They prevent to earlier ex- exunation of the filtration of the soil. In the exchange of the nutritional value of the food, golden rice, vitamin A, or enriched rice, uh, enriched rice. The additional of the using GM has been used to create the taller made the plant and supply the alternative resource in industry the stretch fuels and permeating permeational permeational stress and permeational. Some of the application of the biotechnology in agriculture. In agriculture, study in the detail and the product of pest-resistant plant who could be decrease the amount of the pesticide used, but the toxin is produced by the and but the toxin is produced by the mm, but toxin is produced by the bacterium is called the bacillus of theriogenesis, BT in food short. BT is the toxin gene. BT toxin gene the mm, has been cloned mm, from the bacteria and by the expressed in a plant. They provide resistance to insect with resistant to insect with wood. The insecticide insecticide they affect the create the pesticide एग्जाम्पल हो जाएगा बीटी कॉटन बीटी क्रोन राइस टमाटो पोटैटो एंड द सोयाबीन etc. The BT cotton, some of the strain of the Bacillus thuringiensis, some strain of the Bacillus thuringiensis produced protein that kill the certain insects such as the lipodo, lipidopterinal tobacco woodworm, armyworm, and the coelopatrum. Coelopatrum is a beetles. क्या क्या बोला टोबैको बर्ड वर्म आर्मी वर्म को कोलोपेटलस कोलो कोलोपेटेंस ये हो जाएगा बीटल्स एंड टिप डाइपेथ diphtheria हो गया था less mosquitoes for less mosquitoes. Bacillus thuringiensis from the protein crystals during the particular phase. Their growth. These crystals containing the toxic insecticidal protein. This these crystals contain the toxic insecticidal protein. Why does the toxin not kill the bacillus? Actually, the Bt toxin is the protein existing. Bt toxin protein existing होता है inactive protoxin, but the insect inject the inactive toxin they converted into the active and from the toxin, and due to the alkaline pH the gut which is the solubilize the crystal the activated toxin bind and bind to the surface of the midgut epithelium cell, and they create pore. Thus, the swelling and lysis, and eventually they cause death of the insect. The, the, the specific Bt toxin is the genus whose the specific Bt toxin is the gene whose the isolated from the Bacillus thuringiensis, and the incorporates survival crops and plants such as cotton. They choose the genes depend upon the crop and the target the pest. As most Bt toxin insect group specific, this toxin is coded a gene cry1AC named is cry. There is a number for the protein encoded by the gene cry1AC and cry2AB. Control the cotton uh, wall woolworm. They cry one AB. Control the cotton woolworm that cry one AB. Control prawn. अच्छा sorry. Cry one AC and cry one AB कंट्रोल करेगा कॉटन वॉर्म को, एंड cry one AB कंट्रोल करेगा कॉर्न बोल को cry one AB एंड cry one AC एंड cry one AB ये कंट्रोल करेगा इसको क्या नाम है कॉटन वॉर्म को, कॉटन बॉल वर्म को, एंड cry one AC ये कंट्रोल AB cry one AB कंट्रोल करेगा ये pest resistance of the plant survival is the new pest resistance survival of the nematode pesticide wide variety of wide, wide variety of the plant and animal including the human being. The nematode is the incognate in fact the root of the tobacco plant causes the great reduction in root of the tobacco plant great reduction the unit of The name of the new genus covalent impact the root tobacco plant or the grain reduction field. The novel strategy was adopted the prevent infection which are based on the process of RNA in RNA interference. The RNAi is a place all the eukaryotic organism method to the cellular defense. This method involving the silencing in space mRNA silencing space mRNA silencing space mRNA due to the complementary double stranded RNA molecules bind and prevent the transaction of mRNA. The silencing 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 this source is the complementary RNA could be infected by the virus RNA genome's mobile genetic element transposes that is the replicate by RNA intermediate using the agrobacterium vector. Nematoda specific gene was the introduced into the host plant. Nematoda Nematoda specific gene introduced in the host plant. They introduce the DNA was that production the both sense and antisense was the structure that produced the both sense and antisense RNA in the host cell. There are two RNA being the complementary to each other form the double stranded RNA. The initial RNA is thus silenced the specific mRNA nematode. The consequence was that the the parasite could not be survived in the transgenic host and expressive space and interference RNA. The transgenic plant therefore devote itself to protected parasite. The transgenic plant protected parasite. The biotechnology application is the medical recombinant DNA technology because this process is made of the in impact the area of the health care enabled to enabling to the mass and the production of the safe and more effective therapeutic drug and further recombinant therapeutic does not induce the unwanted immunological response in the common cases is similar product isolated non-human source. The plant is a 13 recombinant therapeutic has been the approved for the human use the world over. In India is the 12th of these the presently being the marketed. The genetical engineering of the insulin, the management of the adult one side of the diabetes possible by taking the insulin on the regular time in the interval. What would be the idea diabetic patent does it enough the human insulin available if they discuss this, they would be relied. One would be the isolated and use the insulin from the animal food. The insulin isolated from the other animal as the effective and thus secreted by the human body itself. It is not elastic elicit immunity elicit immunoresponse. The human body means the bacterium was unavailable. They put the human insulin, it suddenly the whole process becomes so simple. You can easily grow a large quantity the bacteria and make such the insulin need. Nothing about whether the insulin can be orally administered it to the diabetic people. Orally administered diabetic people or not. The insulin was the diabetic it earlier excreted from the pancreas and the cellugated in the crystal and pig. The insulin formed the animal and therefore they through the food and some particles develop allergy and the type of the reaction to the foreign protein. Insulin contains the two short polypeptide chain A and chain B. They linked together in this sulfate bridge. This sulfate bridge in mammals, including the human, insulin synthesize the pro-hormones like coenzyme. Pro-hormones also need the process before it becomes the fully matured and functional hormone. When the fully matured and functional hormones contain the excreted straight called the C-peptide. C-peptide. The C-peptide is not C-peptide are not present in the mature insulin. Is removed during the maturation. Is removed during the maturation. The insulin. The means the changing for the production of insulin using RDNA. Choosing the challenge of the production of the insulin and using rDNA technique was getting the insulin assemble and the mature from the 1983. Eli Eli an American company, prepared the two DNA sequence of the corresponding A and B. Eli Lilly 1983 American company performed the two DNA sequence corresponding to the A and B. The chain of the human insulin introduced then the plasmid. plasmid E and coil the produce the insulin chain. While they produce the insulin chain A and B, where the produced separately, the excreted and combined by the creating the disulfide bond from the human insulin. Disulfide bond from the human insulin. Gene therapy: the person who born with the hereditary disease, the correction of the therapy taken from the such disease. gene gene therapy, in therapy and it attempt to does the gene therapy is the collection of the method along with the correction of the gene defect. Gene defect has been diagnosed child embryo. Here the gene or the here the gene inserted into the person can resell and reduce treated the disease correction. The genetic effect involving the deliver of a normal gene into the individual will rapid embryo take over the function of and compress it and the non-functional gene. The first clinical gene therapy in 1990. 4-year-old girl, adolescence, demain deficiency, adenosine demain deficiency. The enzyme is the uh, CRU crucial for the immune system in the function. The disordered cause due to the deletion of the deletion of the gene, deletion of the gene additions and diminish the children ADA, diminish the children ADA deficiency. Can you code the bone marrow transplantation other by the target by enzyme and the represent the therapy which is the function of adenos adenosine dehydrase adenosine deaminase. Adenosine deaminase gives the patent to the insect, but the problem goes the approach through the non-completed creative. The first step toward the gene is the therapy. A lymphocyte, the blood and the patient and grow and the culture outside the body. The functional the functional ADACDNA, then the introduce the lymphocyte, which is the subsequent return to the patent. However, the cell is not immortally. The patent requires the periodic events such the genetical engineering, the genetical engineering lymphocytic. However, if the gene is isolated from the macro and cell, the ADH introduced into the cell and the earlier embryo stage. It could be permeable parametrical to the molecular diagnosis use effective transmit the disease, the earlier diagnosis and the understand the pathophysio- pathophysiologically very important, and using the conservational method of the diagnosis is serum and urine analysis is the earlier decision the if not possible the recombinant DNA technology, the polymer chain reaction enzyme and link immunosorbent as are the sum of some they survive if the purpose of the earlier diagnosis, the presence of the pathogen bacteria virus etc. The normal suspected only when the pathogen, the production of the disease is disease symptom. This time the concentration of the pathogen already is very high in body. Concentration of the patient already is very high. However, the low concentration of the bacteria and the virus, when the symptom of the disease are not visible. The discharge by the amplification there by the nuclei and by nucleic acid by PCR can explain. The PCR can detect very low amount of the DNA. PCR the rotationally it detached the HIV and separated the ADH patent. It means the use that details the mutation gene and substrated it cancer patent and to the powerful technique to identify means other the genetic disorder. The strand of the DNA and RNA single strand of the DNA and RNA target to the radioactive molecule and allow the hydrate and the complementary DNA payload of the followed the detection using the autoradiography. A clone has been the matured gene, hence the not appearance in the photographic film because they prove when will not be the complementary mutated gene complementary immunity. Ilas is built on the principal antigen antibody interaction. The interference of the pathogen can be detected by the presence of the antigen. The glycoprotein or the detection if the antibody is synthesized against of the pathogen. And trans we are in transgenic animal. The animal can be had their DNA and manipulated the process, the express and the excrete the give the new as the transgenic animal. The transgenic eat rabbit, pig, for transgenic animal rabbit, pig, rabbit, red rabbit, pig, sheep, cow, and fish has been produced the all through all the 95% and all exist in the transgenic animal and the mice. Why they are the animals having the production and how many benefit to the modification. Let us try to explain the normal physiological and the development. The transgenic animals can be specially designed a lot to the study and the gene regulation that affect the normal function of the body and the development that example study the complex factor of the involving the growth such the insulin like the growth factor or whether introducing the gene for the other species formation the factor and the studying the biological effect and the result in information obtained about the biologically remove the factor in the body. Factor in the body, the study of the disease, the many transgenic animals are designed to increase the understanding the gene contribute to the development of the disease. Is there is the especially made of the submodel human disease, model human disease. The investigation of the new treatment for the disease is made of the possible that today is the transgenic mode of exists in the many human disease is such as cancer, the cystic fibrosis and rheumatoid arthritis and eligible the biological production medicine is a required for the treatment of the certain human disease can be contained the biological production and such is the production is not often expressive if for make the transgenic animal such as the produce the useful the biological production can be created by the introduction of the protein DNA code of the particular production such as human being in alpha-1 antitrypsin use the treatment of the emphysema. Similarly, the empathy of the being made of the treatment of the PHENU ketonuria, phenylketonuria, PKU, and the cystic fibrosis 1997. The first transgenic Pauruzi the produce the human protein and enrich milk. The milk is the containing the human alpha and lactate baseless was the nutrient and the more balanced production of the human and baby and normal cow milk. Normal cow milk, the vaccine safe and the transgenic the mice being to develop using the testing the safety of the vaccine before they used in the human. The transgenic mice becomes they use the taste and safety and the poorly vaccine. And if the success and found in the realized reliable, they could be replaced. Use the monkey as a monkey to taste as a bit bit batch batch of the batch of the vaccine. The chemicals after testing, they are known the toxicity, the safest of the toxic taste is they produce the same used the testis and testing that toxicity, the drug and the technically transgenic animal is made that carry the gene which is the made them the more sensitive, toxic, and the substance. The non-transgenic animal there is a there exposed the toxic substance and affect the study of the taxonomic testing such as the animal will be allowed to obtain the resulting base time. The the manipulation of the living organism. The human race do not grow any further without the regulation and the ethical standard and the required to the evolute the morality all the human and activate might help the human and living organism. They go beyond of the morality such as the issue, the biological significance, biological significance, such things, the improvement, the genetic moderation and organism such as the unpredictable result of the organism introduction into the ecosystem. Therefore, the Indian government said the organizations such as the GEAC, Genetic Engineering Approval Community, which is the make the decision of the reading with the um, validity of GM research and the safest of the introduction of the GM organism. The publicly survive the modification using the non-living organism public non-living organism public and survive as the food medical source. For example, all created problem with the pesticide genetic process and they grow in the public arrange the certain complaint being the generated the patient in the produce and the technologies that make use the genetic material plant and the other biological resource used the long been identify the development and use the frames in diagnosis is people the safe region of the region and country. The rice is the important food grain projects which go back to the thousands of the year Asia and agriculture history. There is the is estimated 22 lakh variety of the rice and the India wants the derivative the rice and the India is the richest in world basmati rice is the distinct it the unique aroma flavors and 27 in documented variety of the basmati is grown in India. The refers the basmati is the ancient text and the folklore, folklore and the fruit. It has been grew and the culture in 1997. The American company got the patent the rice, the basmati rice through the USA patent and the tradition make of the allow the company to sell the new variety the basmati US in the abroad and the new variety of the basmati is the actually is derived from the Indian farmer variety. The Indian basmati is the crop semi-dark variety that cleaned and the invasion of the nobility. They are the patent is the extended formation of the equivalent in inflaming the other people, selling the basmati rice and the restrict the patent the survival of the attempt and they made the patent use the produce also process on the based on the Indian traditional of the herbal and the medicine that turmeric and neem which is not the vaginal vaginally intermediately country patent the application the country inevitable is the income our rice legacy and the many able to the anything about the bio bio biovirus is the term is refers to use the bioresource by the mutually company and their organisms without the proper autotherm THR autotherapeutin is the country and the people is the concern without the compin- combinatory plan that the industry of the national rice is financially but pure the blood biodiversity and the temperature in knowledge contract the developing and the undeveloping world of the rice diet biodegradable and the traditionally knowledge the related bioresource that traditionally knowledge related bioresource can be exploited developed modern application can save time, effort, and the expenditure, and thus the commercialization, the brewing, the releasing, and the inject the indicate the competition benefit it share between the developed and the developing country and some nation and the developing law is a culture region such the unauthorized exponential. The bioresource is the traditional knowledge is the India Parliament and the recent clear. The second immediate evident the Indian patent bar issue is the condition including the patent term, the emergency is the prosima and the research, the developing inactivate.